field of neuroscience, while it's rapidly growing, is still sort of in its infancy. It's just like starting to crawl, if you will. And so while we're learning a tremendous amount, there's still a much larger amount that we still don't know.
Ken's goals and the goal of the brain preservation community, some of them are very ambitious and far-reaching. Reasonable people and reasonable scientists can look at it and not agree with all of it, but I think there are a series of things that we should agree on. As neuroscientists, we should agree about the physical nature of memories.
There are plenty of reasons to disagree with brain preservation. There are scientific reasons, and there are sociological reasons, philosophical reasons, and a whole host of other reasons. What I would really like to be able to tell my neuroscience colleagues to do is to make sure that we separate those issues and not conflate them. If I want to understand how a computer chip works, it is incredibly important to have a complete circuit diagram of that chip. The brain is even more important for having that circuit diagram, and I'll tell you exactly why. It is generally accepted within the neuroscience community that the vast majority of the types of memories that we usually talk about are encoded by the strengthening and weakening of synaptic connections.
These words that I'm telling to you now, those are in your short-term memory. And you can remember the last sentence that I said, but very soon that sentence will disappear. And then there's this idea of long-term memory. And long-term memory is very different. It's something that's durable. It's something that can last for an entire lifetime. And we've known since the 60s that long-term memory is completely not electrically mediated. It doesn't depend on it at all. It's a structural and chemical change that occurs in the brain.
And so knowing the circuit diagram of the brain gives you not only the functional architecture of the brain, but it gives you the memories that have been encoded in that brain. In fact, all of the learned knowledge, episodic memories, declarative memories, procedural memories, this is literally written into the neuroscience textbook. The brain is so complex that there might be exceptions to this rule. In fact, I can point to one exception to this rule in the cerebellum where there is good evidence that a single cerebellar Purkinje cell can learn the timing between two stimuli without modifying synaptic connections. Now, everybody thinks that this memory is being encoded by ion channel differences. And of course, ASC preserves ion channels. So this is a non-issue to a certain extent.
So there are folks that are convinced the structure alone is sufficient for emulation. I'm not entirely opposed to that, but I do think there might be another layer beneath which also needs to be preserved.
The most sophisticated criticisms all revolve around what biomolecules are being preserved and which ones may be being extracted and do they have a role to play in memory. And I think the way that you begin to approach answering this is first of all showing what you're able to preserve.
Maybe you would have to map out the receptor density on individual synapses. Maybe you would have to know something about the ion channels on different neurons.
For the next year or two, mostly amounts to characterizing the biomolecular preservation we're able to achieve. We've demonstrated that we can preserve structure, and now the question turns to, can you preserve biomolecules? Can you preserve DNA? Can you preserve mRNA? Can you preserve lipids? Can you preserve proteins? Can you preserve the phosphorylation states of proteins, the glycosylation states of proteins? All of these things that may be relevant for memory. And then you can continue to say we're preserving these different biomolecules, but I think a question people really care about a lot is, can you demonstrate that you've preserved a memory? Can you actually extract a memory from an animal?
We get a few more of these studies showing that all of the things that neuroscientists currently say are implicated in learning and memory are preserved well, then we're going to have to wait a little bit.
The complexity of the overall system might make it extremely difficult in practice to decode memories. We look at it with an electron microscope, we can't see where the proteins are. We try to immunostain, but the glutaraldehyde fixation messes with the immunostaining. We try to interpret it, but we don't know enough about neuroscience to interpret all the little details. What is being asserted here is that we can preserve a brain today in a state that could be decoded in the far future.
There are things that we can do that are closer that I think will demonstrate that that's overall feasible, showing that the structure of memories and the structure of thought can be retrieved in ways other than, say, like running it biologically.
If you were going to test for a memory in an animal with a reversible technology, the obvious way you would test for the presence of a memory is looking at the behavior. You would, train an animal to do something and go through the preservation and see if it can still do that thing at the end. And what I think is a really interesting point is that you can still do something very much like this, even with a preservation technology such as the one that we're using. The way you do this is you don't focus on directly decoding the memory. You focus on proving the memory is there. And it's a little bit of a subtle difference, but thinking about it this way enables you to treat it like a cryptography problem. And there's ways to demonstrate information's there even if you haven't decrypted it.
Today, we have a few studies that have retrieved very simple memories from C. elegans, a very small worm, and from the olfactory circuits in a mouse. What needs to happen is those experiments need to go a little bit further forward into areas like procedural memories and declarative memories. And it's a huge area of neuroscience that's just rapidly advancing.
I'm very optimistic that you can make a validated brain preservation technology that can preserve long-term memories, and I'm optimistic that we will continue the blinding trend of increasing the power of imaging technologies over the next several decades. There's already projects talking about scanning an entire mouse brain. I do think that it's reasonable to look ahead and imagine technologies capable of scanning an entire human brain. I wouldn't want to bet against it.